Hello, good morning. So this is Mrs. Maser again as your teacher in science, technology, and research. Our first topic for today is all about scientific method. Let's do first a simple activity. Actually, have this one on your first learning activity plan as a preliminary activity. So let's perform the following task. Observe your surrounding. Determine arising simple problems like why different plants grow at different rates. And these are the questions that follow. What are your steps on solving the problem? Are your steps derived from a specific theory or discipline, or you just solved it by an instinct or using your instinct? Why is there a need to be organized on solving a problem? So before anything else, so after that very simple activity, let's have first a definition of scientific method. Actually, there are a lot of scientists who have their own definition of scientific method. So, scientific method is a step-by-step process used by scientists to solve problems around them. It is a systematic and organized way of coming up a solution. It can also be used to test whether any statement is accurate. You can use the scientific method to study a leaf, a dog, an ocean, or the entire universe per se. We all have questions about the world, even to what is happening right now. The scientific method is there to test if your answer is correct. You could ask, why do dogs and cats have hair? One answer might be that it keeps them warm. A good scientist would then come up with an experiment to test whether the statement was accurate. So the moment a particular person or even a student tries to test the problem through experimentation and analyzing the data, then that particular person is already doing scientific method. So when we say step by step, it should be organized with validated data to base your conclusion, and the data should or was experimented and tested. So always remember that if you are actually doing a research or trying to answer a specific problem, you need to be specific. You need to test your solution to that particular problem. So during our grade 6 years, and likewise the entire high school years were being discussed with or provided with the information or idea and even knowledge about the scientific method. So we have different steps, but basically we have these steps that I will mention on this particular video presentation. So the first one is identifying the problem, formulating the hypothesis, design an experimentation, record and analyze the results, and lastly, drawing the conclusions. So we'll discuss this as we proceed to our entire discussion. So firstly, in identifying the problem must be clear, specific, can be tested and evaluated. Those are the things that you need to remember when you are trying to identify or research for a particular problem that you observed on your surrounding. Most of the problems are being solved nowadays with the type of technology that we have, with the type of scientific advancements that we have nowadays. Most of the problems right now are easy to solved and innovate likewise. No? So the type of technologies that we have right now, like phones, computers, laptops, are being updated, and new type of model are being released even every year. So it's very easy to solve the problems that we have in our surrounding nowadays. It's just that you need to be very systematic on solving your problem. So for example, why does women with PCOS or polycystic ovarian syndrome, just like me, I actually have PCOS or polycystic ovarian syndrome, is having difficulty bearing a child. So the question is very clear because you have a target, which is women, although you could restrict, you could limit the type of the subject that you will have on your problem. So you could limit that with the type of, with the age. Let's say you're just only focusing on 25 to 30 years old. What are the effects of having a PCOS with that particular age limit? So likewise, another would be diet plan. So what is the effect of having a diet plan if you have a PCOS? Another would be pills. No, what would be the effect? What are the effects of pills if you have a PCOS? No, so you need to be, again, that the problem is specific. You have the subject, the women, the problem itself, which is actually, the polycystic ovarian syndrome, and what would be its effect to women. But then again, on the problem, this particular topic is already answered and actually found on textbooks, even on the internet. Thus, this particular topic, this particular problem is invalid because the answer is already found in textbooks. Next would be, why is there no appearance of butterflies in the garden? Is it valid? Is the problem valid? Yes. This type of question is not found on books. But is the question clear and specific? No. You must state the location. Why? Because the location, like for example, if you have an urban area and rural area, then there will be a great difference between the two. Because in an urban area, it is somewhat polluted, populated, and most of the areas are converted into subdivisions, houses, establishments, which makes an urban area not really conducive for butterflies to live on. Whereas on rural area, you have a lot of trees, still spacious for the butterflies to really have this conducive type of living. So you need to state the location. And then, of course, you need to state the time or the date of the observation, because there are some months of the year wherein butterflies are actually found busking on trees due to heavy rainfall. So on the months of, for example, on the months of June, July, and August, it would be very hard to observe occurrences of butterflies due to there are actually found busking or hiding on trees or on leaves. So you need to be very specific on the time, date, and then the location of the particular subject that they are trying to get involved with your particular problem. So, to revise the question, why is there no appearance of butterflies in the LCT gardens? You need As you can see, it's very specific because LCT Garden at the University of the East, I mean, UE per se is, both UE Manila and Caloocan is found on an urban area. UE Manila is found on the very heart of Manila itself, which is actually populated. And there are a lot of establishments on that particular area. Less trees are observed on that particular area. Likewise, same with Caloocan, wherein you will find the same kind of structure. So, for the month of August, so as you can see, it's very specific. The month is stated. Thus, as you can see on the month of August, the number of rainfall is high. So the location might affect the abundance of butterflies or even the month. Its temperature, humidity, number of rains, etc. might affect the abundance or appearances of butterflies on a given location at a given time. So on your problem statement, you need to be very specific. Next would be formulating the hypothesis. So firstly, what is a hypothesis? During your high school years, hypothesis is defined as it is actually a scientific guess to an answer. It's actually a wild guess to a particular question. So null hypothesis, we have two types of hypothesis. We have null hypothesis and then the alternative hypothesis. The null hypothesis begins with the statement there is no, or it negates the problem statement. Example, there is no significant difference between the humidity in the months of July, August, and September on Sampaloc, Manila. As you can see, the problem is very clear. The months are actually stated. The location, although Sampaloc, Manila is very broad, but you can choose 2 to 3 barangays out of that particular location, which is Sampaloc, Manila. And then the only target is all about the humidity. And then that's it. So the problem is very, very specific. Whereas its alternative hypothesis is that there is a significant difference. So this is actually on the positive side on stating a hypothesis. There is a significant difference between the humidity in the months of July, August, and September on Sampaloc, Manila. So how are you going to answer this particular hypothesis or wild answer to your problem? So you need to go to the next step, which is experimentation. So that's the most crucial part. So firstly, on experimentation, you need to identify first the factors that affect your experimentation. So these are actually your variables, no? So the factors in an experiment that can be changed are called variables. Some examples of variables would be changing the temperature, the amount of light present, time, concentrations of solutions used, etc. A controlled experiment works with one variable at a time. So if several variables were changed at the same time, the scientists would not know which variable was responsible for the observed result. So that's why you need to have a controlled experiment, because in the controlled experiment, you need to set it as your standard. So there will be no changes at all on this particular group or population. So with all the factors that you will consider on your experimentation on this particular group, you need to control it, and there will be no changes at all on this particular population or group. Now, in a controlled experiment, only one variable is changed at a time. All other variables should be unchanged or control, because it's very hard if you're having a lot of variables that you need to consider on a particular topic. So for example, what is the cause of death of a plant on a hydroponic setup? It's very broad because there are a lot of factors that you need to consider on a hydroponic setup. Firstly, the solution of the water, the concentration of the water versus the nutrients that you need to inject with the water. So there are a lot of things that you need to consider. So firstly, on your problem, you need to state first what would be your variable that you're going to test. Now, so for example, on your problem statement, what is the effect of saline solution on the growth and development of plants on a hydroponic setup? That would be good because on the problem you state there the saline solution, so you might have 50%, 100%, or 25% solution, and then you put that on your, as you inject that to your plant on a hydroponic setup, then you observe what would be the effect to your plants. Okay, so very, very specific is that particular problem statement. But then again, you need to have a control setup or a control group wherein you will not inject any type of saline solution. Okay? So an experiment is based on the comparison between a control group and then your experimental group. The control group is very, very important because any changes on the control group versus your experimental group would highly affect on how you analyze your data or you draw your conclusion. So these two groups are identical except for one factor, which is actually your control variable. The control group serves as the comparison or your basis of comparison. It is the same as the experimental group, except that one variable that is being tested is removed. Now, so for example, like the saline solution on a hydroponic setup. So both of the plants are actually on a hydroponic setup. So you have 10 plants under hydroponic setup, which is having a saline solution with different concentrations, of course. Then you have another setup which is actually having the same condition, water nutrient content of the water, except for that it will not have any solution added to the hydroponic setup. So in order to test the effectiveness of a new vaccine, so this is an example, 50 volunteers are selected and divided into two groups. One group will be the control group and the other will be the experimental group. So both groups are given the pill or the vaccine to take that is identical in shape, in size, shape, color, and texture. So both of the groups are given this particular pill. Now, on the control group, even though the volunteers are given identical looking pills, the control group will not actually receive the vaccine. Okay? Now, for the experimental group, this group will receive the vaccine. What variables are kept constant? So, both of the groups are actually having the same size, shape, color, and texture of the pill. Next would be what variable is being changed. Whether or not the pill contains the vaccine. So the control group is very, very important in research. Likewise, in scientific method, because this control group will be your point of comparison on your experimental setup. So as long as you have the control group, you will have the basis on drawing your or analyzing your data. So without the control group, you will not have any point of comparison. So for example, you have plants. No? So you plant oregano plant. It's just that one plant is having a fertilizer. All of the pots, all of the 10 pots that you planted have a fertilizer. It's just that you forgot to have a controlled setup. So the thing is, you will not have any point of comparison on which would be your comparison or your point of comparison. What would be the effect of this fertilizer? So what would be now your point of comparison if you don't have your control setup? Which means your experimental plants, which is actually the oregano, will have no basis at all if the fertilizer is effective or not. So again, having a control setup is very, very important because you will not have any point of comparison if this particular fertilizer is effective or not. Because when you have another plant, let's say the controlled setup, well, let's say you have the controlled setup, but this particular controlled setup do not receive this fertilizer at all. But then again, this particular oregano still grows and develop as time passes by, which means if I am the one who planted the oregano, even if you don't have a fertilizer at all, the oregano will grow and develop. So did you get my point on what is the importance of having a controlled setup? So I hope you have now a bit more clear idea on the importance of having a controlled setup. Next would be variables and non-experiment. So we have two types, the independent and then the dependent variable. The independent variable is the variable that is deliberately changed by the scientist. So these are the factors that are being changed on the experiments. Whereas a dependent variable is the one observed during the experiment. So this dependent variable is the one which is being tested. This dependent variable is highly affected with the things that we do on the independent variable. So for example, on the setup of saline solution on a hydroponic setup, so if as you change the solution or concentrations of solution as that you inject on the hydroponic setup, what would be its effect to the growth and development of the Pechay Tagalog? So which means the growth and development of the Pechay Tagalog is actually dependent on the saline solution, which is actually your independent variable on that particular setup. So the variable should be very clear from the very start of your experimentation, so that it would be very easy for you to take, record, and analyze your data and eventually draw your conclusion. Because if you have a lot of variables that you will consider, it would be very hard on recording because you need to record, for example, on the saline solution, since you're just only testing the saline solution concentrations, I mean like 25%, 50%, and then the 75%, it would be very hard for you to identify if you will add additional variables such as the level of water, isn't it? On the as another factor. Another factor that you might add is the type of plant that you will add on that particular setup. Like for example, you are comparing Pechay Tagalog growth on a hydroponic setup versus bok choy or Chinese pechay. So it is actually very hard. So you need to focus only on one specific target on your problem. Next would be, in the above example, what is the independent variable? So again, on the vaccine that I stated a while ago, that you have 50 volunteers divided into two. Then you have the experimented, which receives the vaccine. And then you have the controlled, which did not receive the vaccine. What are actually the independent variable on that particular setup? And then what are the dependent variable? So the independent variable would be the vaccine that this particular group, even though they receive this particular pill, what would be the effect of this particular vaccine to them as they take the pills. Likewise, the dependent variable would be the effect to their health as they receive both of the pills and then the vaccine. Now, for analyzing the data must be recorded accordingly and should be well organized, so it would be very easy to interpret the data. So you need to make a table so that it would be very clear for you to interpret it. Like, for example, number of days, number of people that will take the vaccine or the plants. So you need to be very specific. And then the growth and development. So on the growth and development, the factors that you need to consider are actually height. Of course, you need to take the weight, because the development, one factor that is considered that a particular plant is developing is that through its weight. And then one more thing is the additional roots or root system. So in a hydroponic setup, the root system is a one good indicator that your solution on that particular hydroponic setup is very, very effective. So the root system is also another important factor to consider or to record on your data. Now, does the data support your hypothesis? From that data, for example, on the plants, on the saline solution, on a hydroponic setup, based from the height, weight, and then the root system, is there a new root system coming from the seeds of the Pechay Tagalog that you embedded on the hydroponic setup? Is there a growth in terms of height per week? Was there an additional height in terms of was there an additional development in terms of height, weight, and root system as weeks passes by? So it would be very easy for you to interpret it. So as you interpret it, on the first week, we observe that there is an increase of height of 0.05 centimeter from its original height. So, which means you need to consider also the original data. The original data are those data that is actually raw or was not given any treatment from your experimentation. So basically, these are the materials or raw materials from the beginning of your experimentation. So any changes from this raw materials that you use on your experimentation, so as day passes by, that you saw something that developed from the plants is actually considered as a data. Okay, so you need to record that. If you wanted to record daily, well, that would be very not so convenient on your part, wherein on a weekly basis, there you will see a very substantial um, effect of your treatment on those plants if you have a very long period of experimentation or treatment on your data. So you need to consider that. Now, after which you get the data weekly or monthly from the factors or from the dependent variables that were changed from your independent variables. Now you are ready to draw your conclusion. So before drawing conclusions, the experiments, again, must be tested 3 times or more before jumping into conclusion to make it valid. Sometimes in research, statistical analysis makes the data valid. So no need to repeat the data 3 times or 4 times. Statistical data analysis on a research makes the data valid. Although this is very hard on the part of the researcher, but of course, this would save time, effort, money, of course, uh, to make another experimentation. So, although it would be very hard to get a statistician, but it will save you much more if you will do a statistical analysis. But on a simple um, problem, repetition of experimentation will do. So if you have any more question, I do hope you learned a lot from my discussion. So again, this is Mrs. Maria Cristina Arneser, your science technology research teacher for school year 2021. Thank you and God bless.